Hi everyone, welcome to our webinar today. Thank you so much for joining, and thank you to our speaker, Dr. Oliver Hartmann, who will be talking to us today about advancing cyclic immunofluorescence through antibody labeling. So I've just got a couple of slides to introduce Proteintech in case you're unfamiliar with who we are. So Proteintech is a leading life science reagent manufacturer. We have a portfolio comprising of 3 core product lines. So Proteintech is the original manufacturer of antibodies and immunoassays, and we have antibodies against over 14,000 human targets, with the goal to create an antibody against every protein in the human proteome. We also have our humankind range of cytokines and growth factors, and all of our recombinant proteins are expressed in HEC293 cells. So this means that they've got the native folding and post-translational modifications that you would expect to see in human proteins. Importantly, all of our recombinant proteins can be manufactured to a GMP grade for use in cell and gene line therapies. And finally, we have our range of Chromotech branded high-quality nanobody reagents. And nanobodies are about 10 times smaller than traditional antibodies, and they combine to their targets with extremely high affinity, which makes them ideal for applications such as immunoprecipitation, super-resolution microscopy, and protein purification. So all of our products are manufactured in-house, meaning that we can keep quality control really high throughout every step of the manufacturing process. We also perform a really stringent validation practice, and all of our validation data is available on our website. We try and perform knockout and knockdown validation wherever possible. All of our antibodies are also available in 20 microliter trial sizes for convenience in testing, and we can offer next-day delivery to many countries across the globe. I'm also excited to say that we have been awarded SciTab's Researcher's Choice Award for the 2nd year in a row. And this is really exciting to us, as this has been voted for by our end users, highlighting how Proteintech is making a positive impact throughout the research community. So we also have a new validation initiative where we are experimentally identifying the exact binding regions or epitopes of our antibodies. And we're using structural and computational approaches to map these binding epitopes onto 3D protein structures. And you can view all of that on our website. So by providing this epitope level insight, we can provide total transparency about our products to our customers, which can then help researchers make better decisions about the antibodies that you select for your experiments. And we also have an immunogen selector function on the website, so you can search for antibodies that have been generated against specific regions of interest. Again, this has all been implemented for complete transparency and to help make reagent selection easier for our customers. And lastly, I just want to highlight that we've built off of the information from our epitope viewer and our product database to create an AI-powered search function called Able. So Able is now completely integrated with our search bar and can provide an AI-generated summary for a comprehensive overview of all of our products. But Able also allows you to directly chat and ask for product recommendations, help with experimental design, and access relevant validation, citation, and educational resources. So for example, you could ask Able, what products do you have for organoid differentiation and characterization? And Able will be able to suggest a whole experimental workflow and the best products for each step along that workflow. This tool is the first of its kind, designed by our scientists with customers in mind, and we are completely in control of the information that powers Able, so you don't need to worry about any responses being polluted by outside sources. And if you're interested, you can access Able just via our website homepage. And with that, I just want to introduce today's speaker. So I'm really excited today that we've got Dr. Oliver Hartmann with us, who is a senior scientific officer and postdoctoral researcher in the experimental immuno-oncology group at the Institute of Lung Health and Immunity at Helmholtz, Munich. So Dr. Hartmann earned his PhD from JMU, Würzburg University, where he studied the development of somatically modified mouse models of non-small cell lung cancer. And his current work combines advanced preclinical models with immuno-oncology approaches to help support the discovery and evaluation of novel lung cancer therapeutics. So with that, I'll just stop sharing my screen and hand over to you. And thank you so much, Ollie.
Yeah, thank you very much for this kind introduction. I'm sharing my screen. which should now pop up for everyone to see. And thank you very much for the opportunity to talk about our cyclic immunofluorescence through antibody labeling in today's webinar. Welcome everyone, and I hope by the end of this webinar I can convince you that cyclic immunofluorescence is a very nice and handy tool to create small proteomic overviews with spatial informations on your tissue samples you get either by purchasing or by doing yourself. And while this picture will now look very overwhelming and very noisy regarding the data, in the end, I hope you can understand how it was generated, do it by yourself, and use it to generate your own research and take out the informations that are in these figures. To start with, however, I would like to get everyone on the same page. So we are now, or I will now give a short introduction on the disease we are predominantly focusing our work on, which is non-small cell lung cancer. And as you can see here, it is not just one disease, but it's 3 different diseases disguised as one. Non-small cell lung cancer is roughly 85% diagnosed in every lung cancer patients. And according to morphology, localization, and marker expression, it can be further subdivided into the 3 major subgroups. Roughly 40 to 50% of all patients are diagnosed with adenocarcinoma. This type of lung cancer arises typically in the peripheral or distal areas of the lung, and AT2 cells, the stem cells of the alveolar compartment, have been identified as the cell of origin for adenocarcinomas. And to identify this type of cancer, usually markers such as SFTPC, NAPSIN-A, or TTF1 are used. The second largest group of non-small cell lung cancer is the squamous cell carcinoma, with 1/4 to 1/3 of all patients diagnosed with. It usually is localized in the central area of the lung and proximal airways, and it is recognized by the marker expression of cytokeratin 5 and 6, or delta NP63 and SOX2. And the last group of non-small cell lung cancer is the so-called large cell carcinoma, and this type of cancer is characterized by the absence of a clear marker expression. Thereby, it cannot be characterized into either squamous nor adenocarcinoma. I hope with this I could show that tumors, or especially cancer, are very complex diseases, and to gain novel insights in how to treat those cancers or understand the mechanistics behind disease progression and cancer aggressiveness, we need tools that give us a large overview about the context the cancer grows in, meaning you need informations of the tumor microenvironment, the adjacent tissue. And here I'm just comparing 3 methods that can give you informations in a large scale of the tissue sample on formalin fixated and paraffin embedded tissue. At first, we have spatial transcriptomics, where we get the whole transcriptome of the cells on a spot level resolution, with often subcellular resolution possible. However, this is on RNA transcripts and doesn't give us information about the protein abundance in those areas. The second method that can be used is image-based mass spectrometry. This is detecting the protein abundance with special elemental tags or antibodies that are tagged that can then be measured in a mass spec approach. We get information about the spatial or morphological context, but it's very limited. And today, as far as I'm aware of, up to 40 targets can be measured with one of the approach, giving you subcellular resolution. And lastly, what I'm showing here now is CISIF, which is the main topic of today's webinar. It's, as the mass spec approach, protein-based with an antibody labeling system. We get full morphology informations out of our FFPE samples. Depending on tissue quality and antibody quality, we can detect between 20 to 40 proteins of interest. This is cycle dependent, and the informations are again subcellular. I think what is very important for most academic labs is the price per sample. And here I have calculated the costs you get per sample for up to 20 targets for CISIF and image-based mass spec. This is not applicable for spatial transcriptomics, since we will get the whole transcriptome with this approach. The price here was calculated by using the mean price of antibodies set to 400 euros for a 150 microliter vial, and a primary labeling kit, which was ordered in bulk. The sample below, or the price below, of 47 to 148 euros is when you use the test sample kits provided by Proteintech. And you can see that even with this test sample kits, the detection of 20 targets on one sample is still several factors cheaper than the mass spec approach with 400 to 1,000 euros per sample, or with spatial transcriptomics with 2,800 to 6,500 euros per sample. The prices for these two methods were used or were calculated by using publicly available costs that are depicted by the core units who will carry out these methods for you. And this is already one of the downsides of Decisive, I have to say, because this method needs a lot of hands-on work after sample preparation, since you will carry out the staining cycles, and they can take up to 10 to 28 hours, depending on how many cycles you want to do. So overall, you will be occupied 4 to 6 days, or even up to 2 weeks, depending on how many samples you want to detect. Whereas in the image-based mass spec and spatial transcriptomics, usually you will send off your prepared samples into a core facility where then the progress is done by you, done by them. However, I now want to show you why Decisive and Decisive staining cycle can be worth your time to get out the most information of your precious patient samples you have in your lab. So usually when we do fluorescence-based image analyzation, we are limited by the microscopes we are using, since they can only detect a handful of fluorophores or, for example, colors per round. And by applying this cycle shown here, we can stain, image thin, and bleach the previous cycle. We can reuse our sample for several staining cycles and thereby get more and more information out of our tissue while preserving the sample. And now we come into the protocol I want to show you with the Decisive cycle. Everything I'm showing here is now how we will do it in the lab. This is focusing on formalin fixated and paraffin embedded tissue. However, with adaptation, the protocol works as well on cryosections as well on 2D cell culture. All the buffers we are used are based on PBS. So this is an important step, since other buffers, the protocol did not work as reliable as within PBS. And imagine now you start with a freshly biopsy of patient samples or samples from animals you get. You will start with the formalin fixation of those samples. Here it is important that you remove blood as best as possible to avoid autofluorescence in your samples later on. And furthermore, you avoid the overfixation of your tissue to not get cutting artifacts later on in the process. In the next step, then, we cut our slices into 2 to 10 micrometer thick sections. Here you have to find the sweet spot for your tissue. In lung, we use usually 2 to 4 micrometers, because the thinner the tissue is while cutting, the more you will reduce the autofluorescence and the refraction of the light in your samples. The downside is thinner samples will lose the tissue integrity earlier on in the cycles due to the bleaching protocol. After the cutting, we then use the standard deparaffinization and rehydration of our samples with a xylene and alcohol cascade. And then we come to the first critical part of the protocol, where we conduct the antigen retrieval. With this technique, we hope to unmask or hidden or altered protein epitopes to make them accessible for the antibodies again. And depending on the lab and on the methods you are using, there are several different methods how you can retrieve your antigens. One is boiling or pressure cooking in certain buffer conditions, or you can use enzymatic ways to retrieve your antigens. For this protocol I'm showing here, we microwave our samples in tris EDTA buffer pH 9.0 for 3 times 5 minutes with the settings high, medium, and low. And the antigen retrieval method is important since we can use up to 20 or more antibodies, and the function on FFPE samples of the antibody can highly depend on the retrieval method you use. So for your approach on this technique, you need to find and establish a retrieval method that works the best for the antibodies you use. This can either be TE buffer, it can be sodium citrate, it can be microwave pressure cooking, or enzymes. If you have established there a routine that works for most of your antibodies, stick to it. And then you can continue after the retrieval with the next step, which is the permeabilization of the cell membrane and the nucleus membrane with a detergent containing PBS buffers, and the blocking of your sample with either commercial available PBS buffers, for example, from Lycor, or just 3% BSA in PBS. The next step then is already the detection with our primary antibodies that we use primary labeled. You can either buy some of those already primary labeled, or you can use the flexible 2.0 kit provided by Proteintech, because this makes you independent on the antibodies you are using and makes it very flexible and versatile technique to use. This works the way that you have up to 6 different fluorophores that can be conjugated on your primary antibodies in a short reaction, covalently binding them to the antibody. And afterwards, you are then quenching the excessive flex linker, thereby reducing the autofluorescence or false positive stainings on your samples. And what I'm usually doing is in our lab that for the establishment of new antibodies, I'm sticking to the protocol provided by Proteintech. So I use for 1 or 2 samples 500 nanogram of primary antibody and link it with a flex linker for 5 to 10 minutes at room temperature. And after quenching for another 5 minutes at room temperature in the dark, I then dilute my antibody mix with the blocking solution and incubate it on our samples either for 2 to 4 hours at room temperature or overnight at 4 degrees. And after counterstaining with DAPI, we can then take the first image. And having now scanned our first TMA picture here, I was quite happy with the staining results because we could already observe differences in the stainings regarding the markers depending on the tissue we choose, showing us that this linkaging with the antibodies is working quite nice and our protocol is working fine. However, on the second glance, we observed that we have a very strong background signal, especially in the green 488 nanometer channel. This autofluorescence, especially in lung tissue, is masking nearly all of our true signal, making this channel barely usable. So for this, we had to find a solution to quench and remove the autofluorescence while keeping the epitopes intact. And the problem is that due to the fixation with paraffin, we have a strong intrinsic autofluorescence in our samples due to the fibers, the keratinization, especially in the tumors, the red blood cells I mentioned previously, and in the lipofucsin. And this just overlaps strongly with the fluorophores we were using in 488, making this channel barely usable for us. So I tried several different quenching protocols, and in the end we came up with a quenching step that is based on Sudan black that strongly reduces the autofluorescence, especially in the green channel, making this now nicely usable for us again and increasing the overall fluorophores we can use per cycle. So adding now this quenching step into our staining protocol, we were now ready to continue with our cycle and try to run the first full lab. So therefore, we had to establish a reliable bleaching protocol that can, after our first staining, remove the fluorophores from the previous cycle, ensuring that we have no bleach or bleed through into the next cycle without damaging the tissue during the process. And there we again adapted several protocols and came up with a very efficient bleaching protocol where we submerge our samples into a buffer containing hydrogen peroxide, sodium hydroxide, and PBS. And then the most important step is to illuminate the samples with a full spectrum light with 6,500 Kelvin and 1,500 lumen for at least 30 minutes to remove the complete fluorescence of the previous cycle, and thereby making the sample ready for the next staining cycle. With this, then, we now put our protocol to the test with a complex staining protocol with in total 20 different markers that are clustered in cell identity, growth and stemness thickness, proliferation, DNA damage, and protein stress for the setup of this protocol. And after we have taken these 5 images during the cycles, we run into another obstacle. Due to the process of staining, bleaching, quenching, and the scanning, we have slightly shifts in the tissue that are either due to shrinkage, to movement, or to very minor rearranges during the scanning process, making the alignment very difficult, resulting in a blurry picture where samples could be misaligned and thereby not be correct. So in order to fix this problem, we added a DAPI staining step after every quenching round to give the microscope the best possible DAPI signal, and thereby the algorithm we use the best way to align our samples properly. And then we adjusted the algorithm nearest neighbor, which we use with WARPy, to overlay our 5 images into one. And since I mentioned now the software, I will take a short detour to show you 3 different softwares that can be used to not only merge your image, but to analyze your image and gain the information you want to have from your multiplexed approach. We have the free, which is QPath. It's a free and shareware-based software that is implemented with ImageJ, and it's based on JavaFX. Due to the factor that it's for free and a shareware, there are already a plethora of plugins you can use and easily install, which makes this a very versatile software to use. However, you need the tutorials and the troubleshooting in YouTube and forums, because QPath might be very easy to learn, but it's hard to master and you can get lost in all the function it has. So in the end, some time and coding might be necessary to get into the software. The next one, the cheap, would be MC Micro. This is as well a freeware, open source end-to-end pipeline. But instead of QPath, MC Micro needs a stronger hardware at your side or even a cluster server to run properly. That's why it's only the cheap, because some investment into hardware has to be done to use MC Micro to its full extent. But then it's a fully automated system that has, unfortunately, no graphical user interface. So you need to know how command line and Docker system works, and you have to set up a bioinformatic pipeline. But if this is all working and you are familiar with the system, you have a very strong very strong tool that can take your multi-channel images and give you the single cell data you want to have. And lastly, we have the expert software from Visiopharm. This is a commercial software which is AI driven and is certified for clinical and pathology use. This is very well polished. It's a kind of fire and forget software where you get the tools for the analyzation you want to have ready-made by the company. However, every tool is behind the paywall, so this might be a barrier for smaller labs to buy into every tool you have. While you don't need huge expertise in the coding part, training or getting training by the company is highly advised. These are just 3 examples. There are many more you can use to get your samples. For everything I'm showing you today, I have used QPath because this is my go-to software where I'm now very familiar in and I'd like to work with. So having now shown you this, now we are coming to the decoding of our 20 markers I have stained in this picture. And this is the same picture as you showed before, but now I have just filtered out the 5 markers used for cell identity. We have in green SFTPC, in yellow NAPSIN A. This is to identify adenocarcinoma. In red, cytokeratin 5. In blue, cytokeratin 6 to identify squamous cell carcinoma. And lastly, alpha smooth muscle actin for the fibroblasts or in the connective tissue of the cancers. And when we now zoom in into the cores, we can see that for the marker expression, we can nicely see the separation in squamous cell carcinoma, where we have cytokeratin 5 and 6 positive plaques here of the squamous cell carcinoma, where we have the purple stain or the pink staining of the fibroblasts here in between, and the remaining NAPSIN A positive cells of tracheas that are still present in the tumor tissue. And on the other hand, we can nicely see here that the adenocarcinoma is predominantly or only positive for SFTPC and NAPSIN A, showing you already that with this tool, we can nicely distinguish cell identity. Here is an example on lung cancer. But we not only stain for those 5 markers, but we as well can have a look on growth of stemness. Here we check oncogenic transcription factors that are upregulated upon oncogenic transformation in lung cancer, namely MYC, JUN, the intracellular domain of Notch1, and 2 stem cell markers, NANOC1 or NANOC and SOX2. And again, we can see that the tumors differ in expression of certain markers or of certain oncogenic transcription factors again. For example, squamous is highly expressing SOX2, whereas in adenocarcinoma, we barely see SOX2 expression, but we can see that the family members of the AP1 gene is strongly expressed in the adenocarcinoma, leading us already or giving us already different information now on spatial level on the tumors seen here. Some people might be interested as well in DNA damage and repair mechanisms. So we now stain for phospho-ATR, gamma-H2X, RAD51, and FANCD2, which as well can be localized on subcellular level. And we can see again that tumors differ already in expression due to DNA damage and the repair mechanisms that are utilized in those tumors as well. One example that is of special interest for us, since we are working with protein stability, is protein stress response in those tumors, just as another example. Here again, we have resolution on subcellular level, and we can see localization of our proteins of interest and the differences in the tumors as well. And lastly, we can correlate all this now with the positive stainings for proliferation markers in those tumors, and then, for example, gain informations on how proliferative state in tumors might interfere with certain pathways we are interested in. And with this, I hope I can show you that sometimes CISIF may be or is worth your effort, because we can get the full morphology information and the spatial context, not just the overall protein abundance. We have a true multiplexed method now on FFPE material with single cell and subcellular resolution. It has a lower cost than standard mass spectrometry imaging, and due to the primary labeling, it's a very flexible tool that you can use. Every coin has a double side, and which I already mentioned, it's a time-intensive method that you use because everything is hands-on. And the amount of multiplexing you gain is highly dependent on the quality of the antibodies you use and how good they can detect your proteins of interest and how well your tissue is preserved during the cycles. So this requires a lot of quality control in between just to be sure that your stainings will work nicely in the end and resulting in the information you want to have and you need for your project. But I hope I can show you that CISIF is a nice method that if you need to know where your proteins are and not just how much. And with this, I'm already at the end. I thank you for your attention, and I'm happy to answer now the questions or receive emails for you if you want to have further in-depth comments or have further in-depth questions.
Thank you so much. That was a really insightful talk, and we've had lots of questions already, which is really great to see. Before we get onto those, I'll just reshare my screen, and I've got a couple of slides for you all. And I'm just going to go over the technology that Ollie was referring to there about the flexible antibody labeling kits. So you should all be able to see that on my screen now. So our flexible antibody labeling kits are a novel FC-specific high-affinity labeling technology. And this is a very specific technology to Proteintech. There are lots of antibody labeling kits out there on the market, but this is unique in the fact that it is site-specific, it's high-affinity, and it means that we can utilize flexible without needing to do some of the more complicated and more harsh treatments that you get with other labeling kits. For example, no buffer exchange is required, and you can use really, really small amounts of antibody. So some of the key benefits of the flexible labeling kits is that if you directly label your primary antibody, then obviously this allows you to do direct staining. So this removes the need for a secondary antibody in any kind of application where you'd be looking at traditional indirect staining. This can also work to really simplify any multiplexing that you're looking to do, because you don't need to worry about the indirect staining from adding the secondary, but also when you are doing primary and secondary staining, you need to make sure and be really careful that your secondary antibody is specific to a primary, and therefore You can't be using primaries from different species, from the same species, because the secondary will bind to all of them. In this case, if you directly label your antibody using our flexible labeling kits, then if you perform that, you can label primaries from all the same species, and then you can do the single stain in one step. So it makes your workflow much easier, and hands-on time is reduced, and also will really, really reduce any kind of background issues that you may see with traditional secondary staining. As well as this, as I mentioned, the flexible antibody labeling kits, because it's not a covalent mechanism of binding, you don't need to worry about buffer exchange. So the antibody that you are labeling can be in any form and at any concentration. So it doesn't matter if you're using an antibody that has glycerol or has BSA or azide, you don't have to remove any of those, and you don't have to buy an expensive PBS-only antibody, like, again, you have to do for lots of other kits on the market. And one really big benefit of our flexible kits, as I mentioned, is that it works with very, very low amounts. So you can label 0.5 microgram of antibody at one time. And that's actually what Ollie mentioned in his methodology, that's the amount that he was labeling for some of his sample stains. So our flexible labeling kits are specific to the FC domain. So you buy them to species subtypes, and they can label up to 8 fluorescent labels. So these include our in-house corallite dyes, as well as FIT-C. And we do have other label options available. So we can also conjugate to biotin, HRP, or you can get custom oligos with this technology as well. No matter which kit you buy, it's a really quick and easy 10-minute protocol. So first, you just have to incubate your antibody with our Flex linker and leave it for 5 minutes. And then you add in a Flex quencher to just help neutralize any of that unbound Flex linker. As I mentioned, it works in the presence of BSA, glycerol, azide, and any other preservatives. The concentration range is between 0.1 milligram per mil to 2 milligram per mil. We do recommend that you label immediately before you use, just because we cannot guarantee the stability of the antibodies that you're using, because this is compatible with any antibody from any supplier. So because we're saying you can use this with other antibodies, obviously we cannot vouch for their stability. So for that reason, we do suggest that you perform the labeling immediately before you perform the staining. And on this slide, I just have an example to show you how you can use it to really simplify that multiplexing. So because these flexible kits, as I mentioned, have really, really high subtype specificity, you don't have to worry about them cross-reacting as long as you do the staining in individual tubes, but you can do this all at the same time. So this is just an example where we're looking at 5 primary antibodies, all raised in rabbit. And it's going to stain for TOM20, which is an outer mitochondrial membrane protein, CD147, which is in the plasma membrane, TDP43, a nucleic protein, Golgi2, which is a cis-Golgi network label, and nucleolin, which is again in the nucleus. And you can see that even when we've used all 5 of these colors together, you can get really, really nice clear imaging and not worry about any cross-reactivity or background. So this is, as I say, a really good way to not only multiplex from different species, but you can also multiplex from the same species. Flexible is not just validated in Cyclic IF or IF, but it's actually validated in a whole host of different applications. You can find, as I said, all of our validation data on our website, but it's been validated in flow cytometry, lots of different types of imaging workflows, fluorescent Western blot, chromogenic Western blot with the HRP conjugate, antibody clone screening, and also oligo conjugate applications. So very quickly, I will just highlight to you our flexible oligo ready kits, which are used for generating oligo flex linker conjugates. So this will enable species-specific and high affinity labeling of antibodies with different oligos. So for everyone who has attended today, you will receive a follow-up email in the next 5 working days, which will have a recording to this webinar. You'll also be entitled to a free flexible sample. So we will send the link out as well with that information. Please be aware that terms and conditions do apply for this, and it is at the discretion of your account managers. And we will also send out a certificate of attendance. So the certificate of attendance will come in a separate email, so just keep your eye out for that. And it might end up in your spam or your junk folder if you haven't received one before. So just have a look there if you are waiting on one. And with that, again, a huge thank you to you, Ollie, and thanks to everyone who's asked questions. I will just stop sharing my screen and we can start going through some of those questions that I can see coming in through the chat there. So starting off, somebody has asked, in the context of Cyclic IF, do you have to use a directly conjugated primary antibody, or can you do traditional primary-secondary staining for this?
So for the first round, you can use the classic sandwich system. But due to the process we use for the bleaching, the antibodies will stay on the sample. We are not removing the antibodies, we just bleach the fluorophores. So for the second round, if you use the same sandwich method, you will recognize the antibodies of the first round. So this will not work. You can use it for tricky antibodies that you can, for whatever reason, not label or give you a very weak signal. So you need the amplifying of the secondary sandwich system in the first round. But from then onwards, you should use the primary labeled ones to avoid false positive signal.
Great, thank you so much. That is a really good explanation for why you can't be using these secondary antibodies in multiple rounds of staining. And you're right, it's all to do with which part of the antibody conjugate complex is being inactivated or versus being completely stripped. Like for example, something like a Western blot membrane. It's a different biology that's happening there. So somebody else asked, would high sample illumination alone be sufficient for bleaching, or do you need the hydrogen peroxide protocol to remove the coralite dyes?
So for us, the peroxide sodium hydrogen buffer with the illumination works the best. There are publications out which just illuminate, which can work, but it takes a very long time and is less efficient. And the longer you leave your sample submerged in buffer or solutions, the higher the risk is that it will detach in the end, or you get like the tissue integrity damaged. So I try to use, so be as quick as possible in the bleaching step to not have as harsh conditions on the tissue. You can try. We have done it twice and I was not happy with the results, so I will stick to the peroxide sodium hydroxide solution. But there are publications out there who just use illumination.
Okay, thank you so much. So a couple of questions about quenching as well. Someone has asked, have you tried reagents like True Black for quenching?
I haven't tried other commercial available like quenching materials because you want to keep the protocol like being able in-house and as like cheap as possible. Yeah, so they most likely will work. I haven't tried them. We used for other methods ammonium hydroxide bleaching, Sudan black bleaching, and sodium borohydride bleaching. So there is some publications out there that sum them up very nicely. And we tested them all, and the best was the Sudan black approach we are using now.
Thank you so much. That's really useful. And there's another comment actually in the webinar chat just about those different bleaching methods. So if you are looking to learn a bit more about bleaching methods, that's a good comment just to look back on so you can find all of that information. So someone has asked, is there a specific kind of fluorescence microscopy recommended for Cyclic IF?
I mean, the better your microscope, the better the result in the end. We have one. So these images were taken with a wide field size axioscan Z1 with a 20x planar objective with 0.85 aperture. So it wasn't even confocal imaging. We have tested it on a benchtop confocal image with only 4 possible colors. And there it's working quite nice as well. But I mean, the more expensive your machinery is, the better it is. The more colors you have, this will highly increase the overall amount of proteins you can stain by reducing the number of cycles you have. So there is the limiting factor is the microscope. So the better the microscope, the better the results in the end.
Okay, thank you so much. Someone has asked, is there a maximum number of rounds that you can send the tissue through the cycle before you start to see significant changes in tissue morphology or antibody reactivity?
So we have done, or I have done 8 cycles maximum. However, the last 2 cycles, I have roughly lost 50% of the course by that time that's why I'm showing here the 5 cycles. There it works very nice. The reason is the quicker you are, the more cycles you can squeeze in, but the longer the samples are like stored in watery solutions, the more of the tissue will detach. And you will have, so we are covering the samples just with PBS glycerol and a cover slip to prevent drying out of the samples during the scanning progress. But the removal of the cover slip, of course, will induce mechanical force on the slide as well. So the more careful you handle the slides, and if you work like 8 or 9 days straight and don't have your slides over the weekend in buffer for storage and continue on Monday, the more cycles you can get in without the tissue. I would assume that so far for me, 10 would be the max if you handle them the most careful. 5 is easy to handle, to 6 without any tissue integrity disturbed.
That's great. Thank you so much. And someone has also asked about if you've got any experience with 2D cell culture for Cyclic IF. Are the cells durable enough for multiple rounds of labeling?
Yeah, so I have done 4 rounds of labeling on 2D cell culture in EBD slides, and this worked quite nice with the exact same protocol for staining and bleaching. There you don't need any additional quenching protocols because the autofluorescence should be minimal.
Excellent. Thank you so much. So that's a lot of the questions we've had on the methodology itself, but you mentioned that you're hoping to get this specific method published later this year. So if people want to find that paper, or hopefully when it's out, is the best thing to do to follow you on? Are you active on LinkedIn or?
Yeah, you can find me on LinkedIn. I have the link for our website in the presentation as well, where you can just click on where you will find me, or just write me a mail.
Brilliant. I'm sure there's lots of people who are in this audience today who'll be really excited to see that when it comes out. So maybe just one final question then, kind of about the sort of application of using Cyclic IF for sort of wider drug discovery pathways. What do you kind of see as the biggest benefit of Cyclic IF over other multiplexing methods?
For me personally, I think the flexibility in the panel I can analyze with the chance of getting not only the information like how much of protein is there or how much of transcriptome is there, but I can see in which cell, under which condition I have the expression of what protein and where does it change under treatment. So not included in the talk, but as you said in the title, we are focusing on immunology and cancer. You can, of course, include in the tumor microenvironment stainings antibodies against certain immune cells, T cells. You can include PD-1, PD-L1 stainings. And with QPath, you can gain these spatial informations out of those slides then to see how under treatment conditions might change. Do you see more infiltrations? Do you see differences in the expression of those? So you get your visual informations, you see alterations in the morphology. By that, you can get quantification of your tissue samples as well as spatial informations and how this changes in the tumor. And I think this combination of all gives you a very good setup for a lot of projects to gain a huge amount of data that you can then follow up.
Amazing. Thank you so much. You've really sold me on Cyclic IF. So I'm sure you've really inspired some of the audience to give this method a go as well, particularly using the antibody labeling that we've highlighted here. So with that, I think we can bring the webinar to a close. Just want to say a huge thank you again to yourself, Ollie, for delivering this talk. And also thank you, as I say, to everyone who has attended today and for asking such engaging questions. And with that, I'll just bring the webinar to a close. I hope you all have a lovely rest of your day.
Thank you and bye-bye.